Hello and welcome to Cognito Key, where we unlock complex topics together. Today, we're diving into an incredible biotechnology that sounds like something straight out of science fiction. We're talking about the ultimate plant photocopier.
Just imagine this for a second. What if you could take your absolute favorite plant, the one with the most beautiful flowers or the sweetest fruit, and make millions of perfect copies of it, all from just one tiny piece of a leaf? Sounds impossible, right? Well, let's explore the futuristic world of micropropagation. So, what exactly is this technique? How on earth do scientists turn a tiny bit of plant tissue into thousands and thousands of thriving plants? Okay, let's dive into this. At its heart, micropropagation is a really high-tech way of cloning plants in a lab. You'll hear the term in vitro, which basically just means in glass, like in a test tube. And the process starts with something called an explant. That's just the tiny sample they take from the original mother plant. And believe it or not, this was first used commercially way back in 1960 by a scientist named Morel to clone orchids. And this little chart here just brilliantly illustrates how powerful this is compared to the old ways. I mean, look at that. Traditional methods are slow, they need tons of space, and you're limited by the seasons. With micropropagation, it's super fast. You can do it in a small lab any time of the year and create millions of clones. It's a complete game changer in speed and scale. The whole process is a pretty complex journey that happens in 5 main stages, and every single step has to be just perfect. So let's start at the very beginning, shall we? Okay, so the first 2 stages are all about creating the perfect sterile starting point. You can't just pick any plant. You have to choose a mother plant that is in perfect health, totally disease-free, and grow it in a controlled environment for about 3 months. Then a tiny piece is carefully cut off, sterilized on the surface, and placed onto a special nutrient jelly. Everything, and I mean everything, has to be completely clean to prevent any contamination. All right, our little plant piece is settled in its clean new home. Now the magic happens. This is the part where we go from one tiny piece to potentially thousands. So, here's the crucial point. The explant is moved to a new jelly that's full of special plant hormones. By tweaking the recipe, specifically by using a high ratio of a hormone called cytokinin to another one called auxin, scientists can basically tell the tissue to multiply like crazy, creating a dense cluster of brand new shoots. So now we have all these tiny little plantlets, but they're not quite ready for a garden just yet. They've been living in a perfect, sheltered lab environment. They need to be toughened up a little bit for the real world. So first, the shoots are moved to a medium with a different hormone, auxin, which encourages them to grow roots. Once they have roots, they're carefully washed and moved into sterilized soil in a greenhouse. This final step is called acclimatization, and it's basically like a training camp for the baby plants. It helps them slowly get used to normal light, humidity, and temperature, preparing them for life outside the test tube. Okay, so we've got this amazing plant photocopier. It's incredible, but what are the real-world benefits? And what about the drawbacks? Why does this all matter? Well, the advantages are massive, especially for things like agriculture and conservation. You can produce millions of clones from a single plant, create disease-resistant varieties, and it takes up way less time and space. You can do it all year round, and you get genetically identical plants every single time. It's been a huge commercial success for flowers, fruits, and trees. But, it's not a perfect solution. It's an expensive process to set up and run, and it requires highly skilled scientists. There's also a big risk that if contamination gets in, you could lose all your plants at once. And that genetic uniformity, that can also be a risk. If every plant is identical, they're all vulnerable to the exact same diseases. So this technology opens up some truly incredible possibilities, from reviving endangered species to designing more resilient crops for our changing world. It really makes you wonder, now that we can essentially photocopy nature, what should the future of plants look like? Thank you so much for watching. If you enjoyed unlocking this topic with us, please hit that like button, share this video, and of course, subscribe to Cognito Keys so you don't miss our next one.